Hello, and thank you for joining for this session. My name is Brian Miner, and I'm the Southeast Wisconsin Stewardship Coordinator for the Nature Conservancy. I'm going to be talking today a little bit about biocontrol for purple loosestrife and share some of the successes we've had here in Wisconsin. I'll start today by giving an overview of purple loosestrife and some of the control options, and then talk about biocontrol along with sharing a couple of case studies of recent successes in the wetlands here in the Mukwonago River Watershed. Purple loosestrife is a wetland plant that's native to Europe and Asia. Like a lot of invasive species in North America, it was brought to North America in ship's ballasts. It's a beautiful plant, so it was widely used for landscaping, especially in the Northeast, and quickly escaped because it has no natural predators native to North America. Purple loosestrife can easily form dense stands of thick mats, can spread rhizomatically and by seed. Each flower contains thousands of seeds that are easily wind dispersed. So it can really take over a wetland and outcompete native plants. It reduces native plant diversity and degrades habitats for many birds and insects that depend on that diversity. There are four basic control options for purple loosestrife. There are mechanical controls such as digging and cutting the plants out of the wetlands. There's chemical control using herbicides and the biocontrol. This chart here on the left shows some of the control options and their effectiveness, and really shows how we use an integrated approach to dealing with purple loosestrife. If you notice on the top left-hand side, we see some scattered individual plants with low density. This is a good opportunity to use mechanical and chemical controls, and we can typically have a pretty good success at eradicating purple loosestrife from a site with that low density. As we get to higher densities, we do need biocontrol to help suppress the plant population. And with small populations at medium density, it is possible still to eradicate, but we really need the suppression action of the biocontrol. As we get to larger populations and higher densities, the chemical and mechanical controls are really, really difficult to try to implement. So the goal really is just suppression using biocontrol. As you can imagine, some of these control methods are really labor intensive. Digging plants out of the wetland and then trying to transport them can be really, really difficult to try to do. Chemical control uses herbicide and typically requires certification and permitting. And anybody who's walked through a wetland with a backpack sprayer can tell you it's really challenging work. And a little less challenging is cutting or cutting flowers off plants, but this provides probably the least effective control option. In the 1980s, the Environmental Protection Agency and state agencies started researching biocontrol options for purple loosestrife. Interestingly enough, this was the first biocontrol program that was designed to protect native plants and animals rather than protecting agricultural resources. There are 4 insects that are approved for biocontrol, 2 beetles and 2 weevils. The beetles are fairly easy to work with, so they're really common and widespread across all areas that purple loosestrife is present. In 1994, Wisconsin started releasing beetles, and the Nature Conservancy started releasing them here in the Mukwonago River Watershed at Lulu Lake in 2000. Both species of Gallerucella beetles have 4-stage life cycle: egg, larva, pupa, and adult. They have 2 generations per year. And the first generation pupates and overwinters in the soil and starts to emerge in May. They may lay eggs by early June and then typically die off after that. The second generation pupates again in the soil in the middle of the summer and then starts to emerge in early July, typically right around the July 4th holiday. So the beetles feed on purple loosestrife during 2 stages, during the adult stage and the larval stage. And during both stages, particularly the larval stage, they can do quite a bit of damage on the plants. Both species of Gallerucella beetles have been very successful in establishing and suppressing purple loosestrife. They're widespread in 30 US states and 8 Canadian provinces. There's been a number of long-term studies done. One in particular in New York State has shown reduced density, height, and flower production of the purple loosestrife plants. The beetles are flying insects, so they can disperse within wetland basins. They disperse with an average of 50 to 1,200 meters per year. And they have been documented to disperse up to 20 kilometers. Although this is rare because they typically won't disperse as long as there's purple loosestrife within a wetland. The state of Wisconsin has some limited statewide data due to differences in reporting, especially in the early years of releasing. But we do have some really good examples of success at a number of sites throughout the state. This site here is up in Burnett County, and you can see photos from 2004 and 2006. After several years of beetle releases, you can tell in the 2006 photo that there's very little flowering going on. The plants are much shorter stature, although there's probably just as many plants. They really are being suppressed by the beetles. I'd like to highlight one case study from locally here in the Mukwonago River Watershed. So this is YMCA Camp Edwards, and in 2013 they recognized purple loosestrife within a fairly diverse wetland that they have a boardwalk it runs through. So they started raising and releasing beetles in 2013 until about 2016. When their program director left, they stopped raising and releasing beetles, and so they haven't released anything there since 2016. So I surveyed the site in 2023, and I found that for a good portion of the wetland, there was still quite a bit of beetle activity, and it was clear that it was still suppressing the plants within at least a pocket of the wetland. So this is a great example that just a few years of releases can result in a self-sustaining population. As I mentioned earlier, we've been raising and releasing beetles in the Mukwonago River Watershed since about the year 2000. For many years, we would get our starter beetles each spring from the Department of Natural Resources, and then raise the second generation in individually netted plants like you see here. Then take those plants out into the wetland, uncover them, and allow the beetles to disperse. So this worked fairly effectively, but it did require a lot of effort on the folks from the DNR to get those beetles out across the state. The Biocontrol program in Wisconsin is coordinated by the University of Wisconsin Extension Program, and the coordinator's goal over the last few years has been really to try to decentralize rearing of beetles. So for about 5 years now at the Mukwonago project, we've been rearing beetles in a mass rearing cage, trying to establish a good population of beetles for partners to come and collect and then release at their own sites. So within this cage here, we have anywhere from 20 to 30 purple loosestrife plants that we take care of. And in May, we go out into the wetlands where we have established populations of beetles, collect about 100 beetles, put them in the tent, and we get anywhere from 750 to 1,000 beetles on an average year. We have had as little as 250 in one year to as many as almost 3,000 in another year. Typically, this program has included digging plants in the spring, going out and collecting beetles from wetlands, and then placing them in the tent, and then releasing them in June to July and throughout the summer. A few years back, folks who've been working at other sites in mass rearing cages started noticing that when they overwintered plants within their cage, they started having beetles emerge earlier than when they were actually collecting them from the wetlands. So they got thinking maybe it's possible to actually overwinter the beetles within the cage and make the whole process a lot easier. So in 2022, UW Extension and the DNR convened a working group to test this theory and see if it's possible to overwinter the beetles within the tents. In the winter of 2022 to 2023, we had 17 sites that tried overwintering beetles. 10 of those sites overwintered in cages, and the rest of the sites had different setups that they were using. There was a bit of variability in the overwintering approaches, but overall we found that there was a high degree of plant survivability. And about 50% of the sites ended up with enough beetles for the 2023 growing season. So it looks like this has some promise. A few sites that tried overwintering beetles didn't have any beetles emerge. And then most sites like ours here in Mukwonago, we did need to supplement those beetles to have a good second generation in the year. So we're starting to look at those results and look through some of the approaches and try to find out reasons why some sites were more successful than others. Here locally in Mukwonago, we started with about 20 to 50 beetles in the tent at the end of the 2022 season. And in early 2023, I only noticed a few beetles emerging. So I did end up collecting about 75 from the wetlands in May. And this ended up being our best release year. We released almost 3,000 beetles to a number of sites within the watershed. And we still had about 200 beetles left in the cage when it was time to overwinter. So really looking forward to the results in 2024 and seeing if this approach really worked well. So the next steps for this project is to continue overwintering in 2024 and beyond. And one of the questions that's come up is whether it's possible the beetles are actually overwintering in the uplands, and if that might change the approach we take within the cages and make it even more successful. Thank you so much for joining this session. I feel like I could talk about purple loosestrife beetles all day. So if you have any questions, I think I'll be around for a question and answer session today, and feel free to reach out to me by email as well. Thanks.